Good morning, friends. So in continuation to the last lecture on hybridization experiment by Mendel, in this lecture I will be discussing you about the dihybrid cross. So how Mendel proceed with the dihybrid cross we will see in this class. So what Mendel did, Mendel also analyzed the pattern of inheritance of 7 pairs of contrasting traits in the pea plant. So in monohybrid cross we see that Mendel considered one trait at a time, but instead of that, or in addition to that, he also studied on 7 different pairs of contrasting characters. So he crossed dihybrids. So what is dihybrid? We have already seen this dihybrid when I was discussing you about the different terminology used in animal genetics. So what are dihybrids? Dihybrids is the plant heterozygous for the allele controlling 2 different traits. So these dihybrids are the heterozygous for the alleles controlling 2 different traits. So Mendel thought that if round seed must always be yellow and wrinkle seed must be green, then he expects that this would produce a typical monohybrid cross, 75% yellow round and 25% wrinkled green. So initially he proceeded with the dihybrid experiment, considering that considering it similar to monohybrid, and he was expecting that 75% of the round yellow plants will be there and 25% wrinkled green, as he obtained in the case of monohybrid cross. But what he observed in his experiment, he observed all possible combination of color and texture. He found that plants with round yellow, round green, wrinkled yellow and wrinkled green, all possible combinations were there, and that too in a particular ratio of 9:3:3:1. So seeing these observations, he observed that each of his 7 traits was inherited independently. He makes some conclusion that each of these traits are being inherited independently, and he gave second rule. Forget about the first or second rule. We have to remember that he gave the law of independent assortment. Law of independent assortment. And what is law of independent assortment? It states that inheritance of one pair of genes is independent to the inheritance of other pair. So it is important to remember that one pair of genes is independent to the inheritance of other pair of genes. Here he is not talking about the allelic pair. He is talking about the pair of genes at particular locus. So inheritance of one pair of gene is independent of the inheritance of other pair. Or we can understand it in different way. Allele of one gene are passed to the offspring independently of the allele of other gene. So he was considering alleles present at different locus. Previously, at the time of law of segregation, he told about the alleles of a single locus, alleles of a gene present in single locus. But now he was referring the alleles of different locus. Alleles of different locus. So he further proceed. Here you can understand it by seeing these alleles or position of these alleles on the chromosome. This is the hypothetical situation. Here you can see this is the allele of a particular locus, alleles of a particular locus, homologous or present on homologous chromosome. So one chromosome is from father, another chromosome is from mother. So here you can see that at a particular locus, alleles of a gene at mother's locus it is capital G, at father's locus it is small g. Similarly for capital R, small r, capital S, small t, capital T. So these are the arrangements of gene in the homologous chromosome. So homologous chromosome having allele for same gene at a specific locus. Now at the time of replication, we know that the chromosome will be split into the sister chromatids, and sister chromatids will just have replication of the particular allele. So sister chromatids of duplicated chromosome have same allele for each gene. So this I have kept here just to understand how chromosomes, how genes or alleles are arranged on the chromosome. So previously at the time of law of segregation, Mendel was talking about these two particular alleles. But in the law of independent assortment, Mendel was telling that this pair of allele is having no any relationship, or these two pair of alleles of two different genes, they are inherited independently. They don't have any kind of relationship. So now if you see diagrammatically, or in the form of Punnett square that we have already seen in the last lecture, I discussed that. Okay, before that, today we know that this rule hold only if the genes are on the separate chromosome or if there is no linkage. So today, at this moment of time, or in the present, we know that the thing is, law of independent assortment will only hold true if the genes are present on different chromosome, or if they are on same chromosome, there should not be any linkage. So what is linkage, we will be discussing in some other class. So here you can see that Mendel took yellow and round seeded plant, capital Y, capital Y, capital R, then green and wrinkled seed plant. So he means at the time of gamete formation, the gametes formed were capital Y, capital R, then similarly small y, small r, then they fused zygote, gametes fused and formed zygote of individual. That individual is heterozygous individual. This F1, this is called as F1. So F1 was selfed. So F1 cross F1. So at the time of F1 cross F1, that means at the time of selfing of first filial generation, you can see, you can make the gametes. We know how to make gametes by the Forkline method, or simply in the Punnett square. Randomly this Y and R alleles of different locus will combine randomly. So how they will combine randomly, and that too in equal frequency or equal proportion. So capital Y will be combined with capital R. Then the total there will be 4 possible gametes. So capital Y will be combining with capital R, 1/4. Then capital Y with small r, capital Y small r, 1/4. Then small y capital R, 1/4, and small y small r, 1/4. So these are the alleles, these are the gametes that will be formed. So in this side it is male gamete, this side female gamete. So he tried to predict the frequency, or he observed how this what phenotype he can get. So he found that total 4 phenotypic classes he obtained. The one was yellow round, green round, yellow wrinkled, and green wrinkled, and that ratio was 9:3:3:1. So in this way he conducted experiment and he obtained this ratio 9:3:3:1. And similarly he obtained the genotypic ratio, that is 1:2:2:1:1:2:1:2:1. So 122, 4, 1, 2 and 1 to 1. This is the genotypic ratio he obtained while conducting experiment for dihybrid. Now comes the role of probability. We can predict, without conducting experiment also, we can predict the frequency of progenies in F2 generation or any generation with the help of law of probability. So in the unit 1, I had discussed in detail about the probability, and different laws of probability. So here we can apply the rules of probability. So it can be used to better understand the segregation and independent assortment. Law of independent assortment allow the new gene combination or genetic recombination. So linkage and recombination that we will be discussing in some other class. Here we have to know that law of independent assortment allows for new genetic combination, that is genetic recombination, and mathematically we can predict the possible combination. So there is certain formula, we will see how this formula has come. The number of possible genotype will be 2 raised to n. Suppose you are having two heterozygote alleles, so the total number of possible, actually it is not genotype, it is phenotype. Sorry, it is phenotype, not genotype. So phenotype will be 2 raised to n, that is 2 raised to 2, that is 4. So here just in the previous slide we had seen that for two heterozygote locus or two locus with heterozygous allele, there was 4 phenotypic class. So total 4 phenotypic class will be there. So here you can see for phenotype it is 2 raised to n, for genotype it is 3 raised to n, for gametes again it is 2 raised to n. So we had seen in the earlier example that gametes, there were 4 gametes, 2 raised to 2, that is 4. Now what is law of multiplication? So with the help of law of multiplication, I had already discussed with you in the unit 1 the law of multiplication, or you can also say it is a product law. So if genes are inherited independently, then what is the probability of phenotypes in the F2 generation? What will be the phenotype in F2 generation if the genes are inherited independently? So it can be predicted with the help of law of multiplication. So what here we will take one example, the same example of that yellow and round and yellow, wrinkle and green. So here you can see, suppose there is round and yellow. These are the probability of, what will be the probability of round phenotype if it is monohybrid cross. So if it is monohybrid cross, the monohybrid cross F2 generation. So what can be the frequency of round? So capital R, small r will be crossed with capital R, small r. So here you can say that there will be 3/4 probability that there will be round phenotype. Similarly for yellow which is dominant. So again for yellow also the probability will be 3/4. So what will be the combined probability? So we know that combined probability, that is also called as AND law. So there you can multiply 3/4 into 3/4, that is 9/16. So here in the previous slide, if we will go to the previous slide, you can see it is 9/16 only. This is 9/16. So 9/16 yellow round. Similarly, you can predict for green round. So what will be the probability of green independently? It will be 1/4. And what will be the probability of round? So again it is 3/4. So what will be the total probability? 3/16. So here you can check, it is 3/16. Again we can see for yellow and wrinkle. So what is the probability of yellow? Yellow is recessive. So 1/16. 1/4. 1/4. Similarly for wrinkled. Sorry. So what will be the probability of yellow and wrinkle? So yellow it will be 3/4. Yellow it will be 3/4, and wrinkled is recessive, so it will be 1/4. So what will be the total probability? 3/16. Here you can see 3/16. Similarly, you can see the green and wrinkle. So green and wrinkle both are recessive. So 1/4 into 1/4, that is 1/16. So like this, you can predict the phenotypic frequency or genotypic frequency for any combination, not only for dihybrid but for trihybrid or tetrahybrid or for oligohybrids, any one based on the law of multiplication, that is the probability law. So in this way we can predict 9:3:3:1 phenotypic ratio among the progeny of dihybrid cross. Now what is the application for Mendel's principle? So the first application already I have discussed, that it is to predict the phenotypic frequency or genotypic frequency of the progenies. So how we can predict? So three general processes are there. First is probability method, we have already discussed. The second is Forkline method, we will discuss in the next few slides. And third is Punnett square method, that also we have discussed in the previous lecture also, and this lecture also in the previous slides we have already discussed. So why this Forkline method is required? So for situation involving one or two genes, it is possible to write all gametes and combine them systematically to generate the series of zygotic genotypes, and further dominance phenomena may be used to determine the associated phenotypes. So if there are one or two genes, it is always convenient to draw Punnett square, and you can write the different genetic gametic combination, and you can produce predict the frequency of outcome of the progeny. But when there are so many genes, if there are many genes, so in that case what we will do, in this method, instead of counting the progenies in square combining gametes, here we tally it in the diagram of branching line. So how here you can see we are considering trihybrid cross. So in trihybrid cross there will be total three gametes. You can see here capital D small d, capital G small g, capital W small w. So it is self with capital D small d, capital G small g, and capital W small w. That means what is that? You are having 3 tall plant, 1 dwarf plant, 3 tall plant, 1 dwarf plant, then 3 yellow monohybrid result we are writing 3 tall, 3 dwarf, and in tall there will be 2 combination, yellow and green. Again in yellow there will be 2 combination, round and wrinkle. So based on this branching, in tall, if you again see, in tall there was 2 combination, yellow and green. So in yellow there was again 2 combination, round and wrinkle. In green also again there will be 2 combination, round and wrinkle. So tall yellow round, tall yellow wrinkled, tall green round, and tall green wrinkle. Similarly for dwarf, dwarf yellow round, dwarf yellow wrinkle, dwarf green round, dwarf green round, and dwarf green wrinkled. So in this way you can predict that 3 into 3 9 into 3 27 tall, then 3 into 3 9 into 1 9 tall yellow wrinkled, there will be 9 tall yellow wrinkle. Similarly 3 into 1 3 tall, 1 green, 3 round. So 3 into 3 9 into 1. So total 9 tall green round. So 9 tall green round. Again 3 into 1 into 1. So total 3 tall green wrinkle. So 3 tall green wrinkle. Similarly we can predict the frequency of different progenies that will be produced in trihybrid cross. So if there are 3 trihybrid, tetrahybrid or above that, then we can use the Forkline method. Again here the Forkline method can be used for test cross. So again this 3 trihybrid cross. So this heterozygote alleles, what is test cross? When heterozygote allele is crossed with the heterozygous progeny is crossed with the homozygous recessive parent. So in this case again the same combination, you can see the ratio will be 1:1:1:1. That is with the help of Forkline method we can predict the outcome of the progeny. Now if you see that here we had I discussed the term test cross. So it is pertinent to know now what is test cross and back cross. What is test cross and back cross? So here I have written that the F1 hybrid is crossed with recessive genotype. If it is crossed with recessive genotype, F1 is crossed with recessive genotype, then it is test cross. But if F1 is crossed with either homozygous dominant or heterozygous genotype, it is back cross. So if F1 is crossed with either homozygous dominant or homozygous recessive, then it is called as back cross. That means all test cross are back cross, but all back cross are not test cross. So this is the difference between test cross and back cross. So why this test cross and back cross means, what is the significance of that test cross and back cross? So with the help of test cross, we can identify the zygosity of the dominant phenotype. So whether the dominant phenotype is homozygous or heterozygous, we can assess by the help of test cross. And back cross recover the elite genotype. Back cross, with the help of back cross, we can produce elite genotype. So here you can see this is the test cross. In this picture you can see the test cross. The dominant genotype is not known. Phenotypically it is dominant, capital Y, but along with capital Y, whether it is capital Y or small y, it is not known. So what we will do, we will cross it with the recessive parent, that is homozygous recessive parent. So here you can see this is the phenotype, gametes produced, capital Y, then unknown, capital Y unknown, and here in the another side it will be recessive Y, small y, small y. So in this way you can see the actual result, that a test cross resulting in the dominant offspring, if all the offspring are dominant, if all the offspring produced are dominant, that means the parent was homozygous dominant. But if it is not dominant, if it is in the ratio of 1:1, that means it was recessive, heterozygous dominant. So in this way we can differentiate between test cross and back cross. Hope this difference between test cross and back cross and its application is clear to you. Again I have written here, it is a cross involving F1 progeny and either of the parent, back cross. It is used by scientists to improve a breed or variety of plant or animal. So with the help of back cross, we can improve the variety of plants or animals. So we will see in the later classes, but I think in unit 3 we will discuss how back cross can be utilized to produce new breeds or improved variety of the animals. Then a test cross, it is the cross involving F1 individual and its recessive parent. So why it is used, we have seen here, to know the unknown genotype. So the phenotype of the F1 progeny is examined to know the genotype. So with the help of test cross, we can test the homozygosity or heterozygosity of the dominant phenotype. So this is the use of test cross and back cross. So in this way, I think I have covered all about the dihybrid cross, and at the last I discussed about the difference between test cross and back cross. So in the next class I will be discussing about the various modification of the Mendel's law or the extension of principles of Mendel's law. So till then you just read out the essentials of this monohybrid or dihybrid cross. Thank you very much.